Hi, so welcome to this lecture on Introduction to Human Genetics. So the idea of this lecture is to bring together people who may have come from a variety of different backgrounds and just give them some very sort of basic knowledge about human genetics so that we're all actually starting on the same page. So some people may have come from biology and they may know pretty much all of this already, although maybe little bits they're not too sure of, and then other people may have come from like mathematics and may not be too clear about what a molecule is and so on. And hopefully what we can do with this is bring it all together so that we're all kind of start together. And then, I mean, everything you hear in this lecture, you're going to be hearing again later on the course. And so, don't worry too much. It's just to give us sort of a taste of stuff, really. On that website there is first the slides for this lecture, we're still recording this lecture, and some much more detailed notes, which will give you where you can go into far more depth as to some of the sort of topics that I'm kind of touching on today. So I think I want to get clear that when people talk about genetics, it could often be kind of confusing, contradictory, and people use the same word to mean different things and different words to use the same thing, and different people will use different words to use different things and will argue vehemently about which is correct and so on. So there's a sort of plethora of ways that people have come to genetics, and that's reflected in some of the problems with the language that people use. So if we think about genes and genetics, there's, I think, kind of at least three different ways that are conceptually very different and which give rise to something. So the first thing we think of as a gene is a bit of information. And if you think of like Mendel's idea of a gene is something which gets passed on from parent to child. It says whether you're green or whether you're yellow, or whether you're short and whether you're tall, if you're a pea stalk, whatever. But it doesn't have any ties to anything else. It doesn't go into anything. There's no account of what's doing that. It's just a thing. Now that contrasts with, in quantitative genetics, the idea of genetics, the gene can be pretty much forgotten, or there's no notion of underlying biology or anything. You can just be talking about mathematical concepts, and you can do a whole load of maths and a whole load of statistics without having any kind of thinking about what's driving all of that. And then, especially lately, perhaps, we've had a clearer and clearer biological understanding of what a gene is. And often we think of the word gene to be as it's meant in biology, and then we run into problems if people are using another way. So just to give an example of that, it's common to say that somebody has like the gene for breast cancer. They've got the gene for breast cancer, or the gene for Huntington's disease. We've all got the gene for Huntington's disease. What we mean when we say we've got the gene for Huntington's disease is that there is a gene which, if there's a particular variance in it, particular mutations in it, if it's wrong in some particular way, then that will result in the disease called Huntington's disease. We've all got the gene. And when we say we have the gene for disease, really we're using that in the sense of the information rather than the actual biology. Well, what I'm going to do now is going to really focus a lot on the biology, make sure we've all got a real sound grounding of the biology that underlies genetics. And so we are going to just get a bit of clarity about these very basic concepts of chromosomes, meiosis, mitosis, genes, and then genetic polymorphism, genetic variation, which is kind of what we might be interested in. So the first thing to think about is chromosomes. And a chromosome consists of two strands of DNA. So DNA is a polymorphism of DNA bases, and not a polymorphism, a polymer of DNA bases. And what a polymer is you have lots of very small molecules which are covalently bonded to each other in a big long chain. And we give them symbols A, T, G, and C. It's adenosine, thymine, guanidine, and cytosine. And So there are covalent bonds between what we call the DNA bases, the nucleotides, and they form a long chain, and that's one strand of DNA. And then opposite that is another strand of DNA, and the two strands are sort of held together. They're held together by hydrogen bonds, which are weak bonds, and so they can separate. And we'll see later there's key functions of the DNA. First off, to be able to stay as a double strand, but then also for the two strands to be able to separate. And the DNA code, the genetic code, is based on the sequence of those bases. So we read off there AATACGC, whatever that says. That's the DNA sequence, and we'll see how that code turns into genetic code. And then a key feature of the DNA bases and the strands is that one strand runs in one direction, if you like, the other strand runs in the other direction. Is called it's complementary. And what we mean by complementary is that everywhere there's an A on one strand, there's a T opposite, and everywhere there's a G on one strand, there's a C opposite. And what that means is the genetic code is in both strands. You can use one strand to reproduce the other, and all the information is contained in both strands. Now, there are the thing to bear in mind about DNA. We think of DNA as these sort of X's with a coming together, and they're held together, the ends of the telomeres, the bit in the middle called the centromeres, and then the telomeres again. That is not how DNA usually is. In a cell, a DNA is not in that shape. A DNA is a long, stringy strand of double-stranded DNA, squished up like a kind of mess of spaghetti and crammed into the nucleus of a cell. And it only takes those kind of characteristic X shapes during cell division, during meiosis and mitosis, as we'll see later. So you've got this long, stringy, double-stranded DNA in the nucleus of the cell. Now, there are 23 pairs. They're called homologous pairs. So you have 2 chromosome 1s, 2 chromosome 2s, all the way up to 2 chromosome 22s, and then either 2 X's or an X and a Y. And you've got one member of the pair from each parent. So I've got 1 chromosome 6 from my father and 1 chromosome 6 from my mother, and we'll see how that works out later in meiosis. So that's the sort of chromosomes that we have in our cells. Now, a key feature of thinking about genetics is the process of meiosis, which is the basis of inheritance, of how genetic characteristics get passed on from a parent to a child. And key to that is the process of which forms the gametes, the egg cells and sperm cells. And what I'm going to just show you is what sort of happens to the chromosomes in the process of the production of these cells. What we're going to see is that although so-called somatic cells have 2 copies of each chromosome, 1 paternal, 1 maternal, when we produce the gamete, it's only going to have 1 member of each pair rather than 2, and that's called haploid rather than diploid. So I'm just going to go through this process of meiosis. There are these key stages: duplication, crossover, and then the daughter chromosomes going into a gamete. So the first stage is meiosis. So what we see here is that the 2 DNA strands are separating. So here's your double-stranded DNA. It's magnified here. You can see the complementary bases opposite each other, and the strands are separated. And one strand is used as a template to build up complementary sequence for itself, and then the other strand is also used as a template. They go obviously going in opposite directions. They're built up in opposite directions. But what you can see is that if you do this process down the whole of the chromosome, you will end up with a copy of the exact copy of the chromosome. So you start with 1 double-stranded chromosome, and then you'll end up one length of double-stranded DNA, and you end up with 2 lengths of double-stranded DNA, which are both exact copies of the original. So here is your duplicated chromosome. So this might be chromosome 6. I might have got this chromosome 6 from my father, this chromosome 6 from my mother. And then what happens is, a key feature is the 2 members of the homologous pair physically come together, form a small number of crossovers, exchange genetic material, exchange segments of the chromosome, and then they separate, and that makes 4 daughter chromosomes. And each of the daughter chromosomes, you can see, ends up with as being a mixture of paternal and maternal chromosomes. But what you'll see is that there aren't very many crossovers. So what you have is huge chunks of chromosomes that have been inherited intact, and that will become important later when we talk about linkage. You've got 4 daughter chromosomes. Then what happens is one of those goes into each of these cells. This could be a nucleus of an egg cell or a sperm cell, but either way, you've ended up in a situation that the nucleus only has half of the DNA that the parent had. You've only got 1 copy of each chromosome instead of 2. And then what will happen in fertilization is that the nuclei of the sperm and the egg will fuse, and then you'll end up with your two copies of each chromosome. You'll end up with all your pairs again. That's called a diploid nucleus. And then thereafter, that's the egg that's fertilized. Thereafter, cell division will be by mitosis, and in mitosis, the pairs are both copied and both inherited, so that the two daughter cells are diploid. They have exactly the same chromosomal complement as their parent. And then what we have is that through all the cells of our body have exactly the same chromosomal complement. We've got 46 pairs of chromosomes, which are absolutely identical between all our cells, deriving from this one fertilized egg cell. So that's the process of meiosis that makes the gametes. Now, genes, kind of when people talk about genes, you sort of think about, I don't know, a lot of people, you think about inheritance. You think about genes are the way that things get inherited down from parent to child. And that is true, but that is not really what genes are doing most of the time. So what genes are doing is it's nothing to do with inheritance. They're in the nuclei of every cell of our bodies, and they are working away all through the day making proteins. That is the job of genes, is to make proteins. And then, yes, they're able to be inherited, and then the offspring can make the same proteins as the parent. But what the genes are doing on a daily basis is they're making proteins, and the way they're making proteins involves these stages of transcription, splicing, and translation, which I'm going to go through now, so we've got a clearer understanding of how genes kind of relate to the phenotype, the things that we're interested in people. So you have these long stretches of double-stranded DNA, which is the chromosome, and then some of those stretches are genes, and the genes are actually make up a very small proportion of the DNA in chromosomes, about 1%. So if you say there's 3 billion base pairs in the whole human genome, then 1% of them are genes, are going to be coding, and so that's 30,000 base pairs. So this is the way a gene is right now. If you look at a bit of DNA under a super powerful microscope, which we don't have yet, then the genes do not look any different from the other bits of the DNA. You don't see anything different. So everything I'm showing you here is simply defined by the sequence of DNA bases, which are going to be interpreted by cell biology as kind of genes. So a gene consists of exons separated by introns, and exons are the bits of the gene that are going to get put into RNA, and then it going to end up being translated. I'll show you later. So, and then the introns are not going to end up into in the RNA, and they're not coding for proteins at all, and they can be very big, and there can be lots and lots of exons in a gene with the introns in between them. And then that's kind of the important bit of the gene, but we could also note that there are bits on the DNA nearby which can affect the expression gene, where the gene gets turned on or turned off, where the protein gets made from it, and these are promoters and enhancers and repressors, and so they're a bit. So what that means is that variation in the DNA near the gene can have effects on gene expression. But the main sort of bit, the core bit of the gene, as I'll show you in a moment, is making proteins. So the first stage of producing protein from the DNA code consists of transcription. And what we do in transcription is we use the DNA as a template to produce a length of RNA. So RNA differs from DNA in that the base is, instead of a thymine, you have a uracil, which is coded as a U. But otherwise, you can build up complementary RNA by putting a G opposite every C, a C opposite every G, a U instead of a T opposite every A, and an A opposite every T. So you can build up a stretch of RNA that has the same information content as the original DNA. And in order to do this, what you need to do is the two strands of DNA temporarily separate, and then one of them acts as a template where you can build up a complementary strand of RNA using an enzyme called RNA polymerase, and that will extend over the length of the gene. So the next thing that needs to happen is that we need to take out the bits that were the introns. So the bits of the RNA that consider introns are removed, and you're left with only the bits of RNA that corresponded to the exons, and that process is called splicing. And so conceptually, I've shown you this here. Now, in reality, you get a stretch of messenger RNA, and there's nothing actually different to see here. I'm just marking those joins there, but that's showing you where the bit that was complementary to the introns has been spliced out. And then you end up with a length of messenger RNA, which is going to be used to translate into a protein. So the way translation works is that the code of the RNA gets read 3 bases at a time, and then those 3 bases code for an amino acid, and a polypeptide chain is built up based on the RNA code of a string of amino acids that are covalently bonded together. And this is essentially what protein consists of. It's called a polypeptide is a polymer of amino acids. There's about 20 or so, 23, I think, amino acids, and there are 64 different possible ways to get 3 DNA or RNA bases, because each one is 4 to the power of 3. And so what that means is that there's redundancy in the genetic code. What that means is that you could make a change in the DNA, which will produce a change in the RNA, but it might be that change produced a codon, a triplet, which still codes for the same amino acid. And then that would be called a synonymous DNA change, a synonymous change, because although the DNA, the code has changed, it hasn't changed the amino acid sequence that you get. By contrast, if you make a change in the DNA sequence, which gives you different RNA, which does code for different amino acid, and that's called a non-synonymous change, and you'll end up with a different amino acid sequence than you would have had if you hadn't had that DNA change. Okay, so this is the key of the genetic code. You decode 3 bases at a time, and you build up an amino acid sequence based on those 3 bases. But that's not the end of the process of building a protein, because a number of things can then happen to that polypeptide change afterwards. So you can change how it gets configured in 3D space. You might make disulfide bonds between 2 amino acids that sort of hold the thing in place. And often a mature protein will consist not of just a single polypeptide chain, but a number of polypeptide chains which have come together, and we can call those subunits. And what that means is, it's easy to imagine, each of these subunits will be coded by a completely different gene, maybe on completely different chromosomes, and all going to form the same eventual functioning protein. Now, what that means in terms of disease causation is you might have a mutation in the gene for this subunit, or this one, and either way you'll still end up with a badly functioning protein. So what that means is that variants at all sorts of different places in the genome might damage the same protein, might end up with the same disease, and there's plenty of examples where that kind of actually happens. And also, just to get the sense that there's more that can control, what a protein's like, whether it's working or not, how much of it there is, how well it's working, than simply the genetic code. The genetic code has an influence on that, whether the gene's expressed or not, whether you've got the protein there or not, what the sequence of the amino acids are or not. But lots of other things in the mechanism of the cell also have effects on whether that protein is working or not, where it is in the cell, whether it's switched on or switched off, and so on. So there's more to protein function than just the gene, but the genes have a big effect on it. So what we're kind of interested in when we look at genetics is, although human beings are 99.9% identical, there are some variations between them, and we're kind of interested in the way that variation can have an impact on susceptibility to disease or other traits. So one way that polymorphism is important and useful and interesting is to see how it affects the phenotype, risk of disease. But there's also a completely other way, I think, of thinking of polymorphism, which is important to bear in mind, which is that you can use a variant as a marker. So if you have some variation in the genome somewhere and you can detect that variation, then that variation is tagging a particular place on a particular chromosome, and as I'll show you in a moment, you can use that for genetic markers in genetic mapping studies, association and linkage studies, even if that variant has no effect at all on the phenotype. So 2 different kinds of variants, if you like, variants which affect phenotype and variants which can be used as markers. And to thinking about how a variant can affect phenotype, so the kind of most obvious thing is that you can have a DNA variant in the coding sequence itself. So then that might mean that you end up with an amino acid change, and then obviously that can affect the functioning of the protein, and then that might make you more likely or less likely to develop the disease or have some effect on functioning. The variant may produce an amino acid change, but it might be a trivial amino acid change, so the substitution doesn't make any particular difference to the protein function. Or, as we said, there may be a variant in the sequence But if it's synonymous, if you end up with the same amino acid, then again, you're not likely to see any effect on the phenotype. Then you can have variants, as I said, near the gene, but not in the gene itself. And if those variants affect the machinery of the cell, which is trying to switch the gene on or switch it off, then you can end up with too much of the gene being expressed or not enough, or the gene being expressed in the wrong cells at the wrong time of development or what have you. So that can cause problems. So you can, the point is that the variation outside the gene itself can sometimes have phenotypic effects. And then variations in splice sites can cause very important effects as well. So if you imagine that the DNA code along here is telling the RNA handling apparatus that this is a change from an intron to an exon, and this bit of it's going to be the RNA is going to be snipped and it's going to be spliced out. If this sequence changes here, either in the exon or in the intron close to the splice site, then that splicing may go wrong, and then you can end up with very severe consequences, because you can end up with an intron spliced into your messenger RNA, which is not really coding for anything useful at all. And just while I've got this picture up here, another kind of very severe variation is that if in your DNA sequence you have a coding change which changes the code for amino acid from the code for amino acid to a stop signal. So there's a sequence of DNA bases which is recognized as saying this is the end of the protein stop. If you get a stop code produced, then that will mean that you have a very truncated product, and usually that will just be completely eliminated, but you'll have essentially no functioning protein at all being produced. And the other very damaging sequence variant is if you have one or two DNA bases inserted or removed from the sequence, then because you're reading the DNA bases 3 at a time, if you take one out, then you've changed the reading frame, and then you start just getting nonsense, because the amino acids that are coded for have nothing to do with the ones that you really wanted in the first place. So that's called a frame shift insertion or a frame shift deletion, and that is expected to be extremely damaging for the functional protein. You're expected to get sort of a complete nonsensical, non-working protein. If, on the other hand, you have a little insertion or deletion which is a multiple of 3, so if you put in or take out 3 or 6 bases, then what that means is you just end up with an extra amino acid, or one missing, or 1 or 2 missing, whatever it is. And so that's called an in-frame insertion or deletion, and that's much less disruptive. So the most disruptive variants that you expect to see are stop variants, splice site variants, and frame shift variants. So I just want to mention something about DNA markers before we get back to disease, and show you how they're used for mapping, and a slight bit of history really. So the first DNA markers that we had, which was meant that we could recognize a variation in the DNA which had no effect on the phenotype, were called RFLPs, restriction fragment length polymorphisms, and they were just single DNA based changes which had no particular effect, but which could be recognized by bacterial enzymes. And there was a few hundred of those scattered around the genome that we used for mapping studies. Then the next set of markers that we used were microsatellites, and this was a bit where you'd have a bit of DNA sequence which might be repeated several times. So it might be 2 bases, CA, 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 and that would be repeated, but the number of times it would be repeated would vary, and that meant that you could count the number of repeats that you had, and you could use that number as a sort of tag and be able to, as I'll show you in a moment, follow that fragment of DNA through pedigrees and so on. So there's several hundred of these, and because you had a lot of variation in how many repeats you'd have, they were very informative. And then what's been used lately in genome-wide association studies are variants called SNPs, single nucleotide polymorphisms, and again that's a single base changes from one base to the other. They're all over the genome. There's millions of them. There's a standard panel of about 1 million, which is frequently used. But I've said they're uninformative because you've only got one base or the other base. It's not like a microsatellite where you've got a lot of variation in the number of copies that you have. So they are useful, but they're useful in slightly different ways. And I just wanted to show you this, and also to show you how we get the the variants. So there's a particular technique which is worth knowing about called the polymerase chain reaction, which is used in all sorts of situations to amplify a sequence of DNA. And I just thought I'd put this in here. It's used for detecting microsatellite polymorphisms, but you can use it for all sorts of things. And I thought it's worth just taking you through the method so that you're kind of aware of it, as it's kind of so widely used in so many applications. So PCR, polymerase chain reaction, consists of denaturation, annealing, and elongation, and then it's a cycle, and you just do that lots of times and you get lots of DNA, and I'll show you how that works. So the first step of PCR is denaturation. So you start with this double-stranded DNA and you do something to it. You have a reaction condition, you put it to a particular temperature, which means that the strands separate. So then you have the two complementary strands, and they're separated, and there we go. That's denaturation. Then you have a process called annealing. What you're going to do is you're going to put in oligonucleotide primers, and oligonucleotides are short polymers of DNA, and you choose the sequence of those to match two places on your DNA strands that are fairly close together. One prime site, binding site, will be over here, and one primer binding site will be over here. And what we're going to end up doing is we're going to end up amplifying the bits between the two binding sites. So this oligonucleotide has found a bit of DNA that's complementary to it here on one strand. This has found a bit of DNA that's complementary to it on the other strand, and that is called annealing. The next step is where the polymerase comes in, elongation. An enzyme called polymerase starts with these primers which are bound to single-stranded DNA. It takes nucleotides which are present in the reaction solution, and it extends the primer using the other DNA strand as a template to add complementary bases to this primer. And that means that you get extension along here, and you extend beyond the site of the other primer site. And so that is elongation. So now we've got a new strand of DNA which includes all the bit that we're trying to amplify, and alongside the original strands. Now what we do is we do denature again. We're going to denature these, and we're then going to put in the primers, and we're going to go here. And now when we elongate those primers, what you see is this one elongates to here, and then it stops. There's nowhere else to go. That one can carry on, that one can carry on, but the next one, let me just go, stop. So here, from now on, every time we repeat this, what we're going to do is we're going to get more and more copies of just this DNA stretch, which is between the two primer sites. And you can do that lots and lots of times. You start with one DNA molecule, you multiply up, it goes up exponentially. It's the basis of all kind of sensitive tests for detecting very tiny quantities of DNA, and you can use it for all sorts of things. So if you've seen the movie Jurassic Park, what they did was they got a little bit of dinosaur DNA, they multiplied it up, and then they got loads of dinosaurs. So it works for all sorts of different applications. And as I said, you could use PCR. PCR was what was used for typing microsatellites, but it was also lots of things. So why are we interested in genetics for human disease? What is the value of genetics? So coming from a position that we're trying to understand disease, we can do all sorts of experiments. We can look at things that are wrong in disease people. We can see what's different. We can try to look at predisposing factors. We can look at epidemiology. But the key value of genetics in human disease, understanding human disease, is that whenever you find genetic variation associated with a phenotype with a disease, then you know that if there's an association, if there's a causative relationship, then it's the variation which has caused the disease, and it isn't the disease that has caused the variation. So there's no disease that will produce variations in your DNA code to match the disease. And you can contrast that with other kinds of studies. So if I see a bunch of information in somebody with heart disease, I don't know whether the information caused the heart disease or heart disease caused the inflammation. This thing about causation only applies to the DNA sequence. So if I'll just get in here, it doesn't apply to expression studies or epigenetic studies. So if you look at epigenetic modifications where the DNAs get methylated or whatever, then you don't know for sure whether the methylation gave the disease or the disease gave you methylation. If you look at expression studies, if you see more RNA expression, more protein in a disease than not, that doesn't mean you don't know whether the additional expression has caused the disease or the disease is driving additional expression. But if you look at the DNA code, then potentially you know that the disease did not change the DNA code. If you can find variations in DNA code which are associated with disease risk, then you know the direction of causative effect, and hopefully that will give you insights into what RNA is being produced, what protein is being produced, how's the protein being affected by this DNA code, and you've got a chance to get a better understanding of what the mechanisms of disease, and hopefully then maybe what you might do to produce better therapeutics for it or whatever. So the direction of effect is key. So I want to show you a couple of approaches to mapping disease genes, which is just kind of like important to be aware of. You can argue about whether linkage is particularly relevant there or not, but I think it's important that you need to know about linkage in order to understand other stuff about human genetics. So the two things I'm going to go to show you is linkage analysis and association analysis. And association analysis is conceptually the simpler one. So supposing we've got a bunch of people with a disease and we genotype them for a marker. So a marker is something which varies. There's allelic variation, particular sites on the genome, and we know where that marker is. And we can detect that variation. We can say which alleles somebody has. And we can label the alleles. So this might be numbers of repeats of a microsatellite, and we can label them A to D or whatever for different numbers of repeats. People have got 2 copies of each chromosome. So at a particular point on the chromosome, they'll have 2 alleles, and those 2 alleles may be different or they may be the same. If they're the same, we say the person is homozygous, so they're both the D allele on both chromosomes. If they're different, A and D, we say they're heterozygous. We can take a bunch of people with a disease and a bunch of normal controls, and we can see which alleles of this marker the disease people have and the non-disease people have. And what we can see here in this sample is that the D allele is commoner among people with the disease than among normal controls. So then we can say that the D allele is associated with disease. We can say that the D allele is commoner in people who've got disease, and we can also say that people with the D allele are at higher risk of developing disease. So you can put it either way around. Either the D allele is commoner in people with disease, or the disease is commoner in people with the D allele. But either way, it's a fairly simple statement to say that the D allele is associated with increased risk of the disease. Then we can contrast that with linkage analysis. So with linkage analysis, we've taken a pedigree of people with disease. The squares are males, circles are females, and we're going to genotype everybody. And what we can see in this first pedigree is that everybody who inherits this A allele from their parent gets the disease, and everybody who doesn't. So you've got these 2 alleles. You inherit one from your father, one from your mother, and if the one here that you inherit from your father here is the A allele, you get the disease. This person inherits the A allele and gets the disease. This person inherits the D allele and doesn't get the disease, A and disease, and so on, and then that goes down through their children. So this person doesn't inherit the A allele, they don't get the disease, and this one does and does, and that all goes through fine. And then what you can see here is that this person has inherited an A allele but not the disease, but they didn't inherit the A allele from their father because they've inherited a C allele, and the mother doesn't have a C allele, so they must have inherited the C allele from their father, and this A allele has actually come from their mother. So it's this A allele on the disease, this A allele disease, and so on. So that goes all the way through this pedigree. Anyone who gets that A allele gets the disease. Anyone who doesn't. In this pedigree, it's a bit different. Here it's the B allele that goes to a disease, B with disease, not B with disease, B with disease, not B with disease, and so on. That all works fine, not B, not B. And then this person, it goes wrong. So this person inherits not the B allele, they inherit the C allele, but they still get the disease. And we're labeling them as a recombinant. And then thereafter, this C allele they've inherited goes with a disease, and not C and disease, C and disease, C and disease, and so on. So what may be happening? So if in this original founder here, there's a disease mutation which is giving you the disease in a gene, and nearby there is a marker allele, and that marker allele happens to be an A allele. So you've got an A allele on the same chromosome as the disease mutation, and then they get inherited down through the rest of the pedigree. So and because they're close together, they tend to get inherited together. If you remember that picture of meiosis, you've got big chunks of paternal and maternal DNA being inherited together. So either you get the DNA which has got the mutation on it and the A allele, or you get the DNA from the other chromosome which has a normal copy of the disease gene, no mutation, and whichever marker allele happens to be on the other chromosome. And that's what's going on in this this left-hand pedigree. In the other pedigree, we may start with a different situation. So here in this founder, it may be that they've got the disease mutation and the marker allele nearby happens to be a B marker allele, and then the B and the mutation get inherited down together as before. And then occasionally, although it's rare for two loci which are close together for a crossover to happen between them, it can happen. And so when that happens, you get a recombination. Then the marker allele, which was on the other chromosome, ends up on the same chromosome as the disease mutation. So here it's the C marker allele has ended up on the same chromosome as the disease mutation. And then from now on, everyone who inherits that chunk of chromosome, the C and the mutation, will get the disease, and then everyone who gets the other chromosome will be fine. So that may be that's kind of what's happening. So what we see is we see the markers. We don't see what's happening with the chromosomes. We can follow the markers through pedigrees, and we can use clues like this when a marker allele and a disease happen to be inherited together more than you'd expect by chance. That's telling you that the marker polymorphism is near to a locus which is causing disease. So these methods were using linkage analysis was the fundamental way in which the map of the human genome was built up, and was also used to localize genes for many Mendelian diseases. So it was a very kind of effective method for localizing disease genes. Now, there's a few things I want to contrast between linkage and association. So one thing is that association is a high risk allele, so you say association with the D allele. In linkage, there's no high risk allele, you're just saying there's linkage to the locus. So it's here it's the A allele, then the B allele, and it's the C allele. So it's not, there's no high risk allele with linkage, it's just with the locus. So the next thing to say is that association studies you'll sort of broadly carry out now of unrelated people. I mean, what we'll see later is that they'll be they're very distantly related, but you basically take a sample of unrelated people and you see whether there's association. Whereas in linkage, you're always doing linkage between people who are related in one way or another. Maybe the relatives in a consanguineous family, but one way or another, you're relying on relationships in order to detect linkage. And then the third thing to say is that association is a very short-range phenomenon, and linkage is a very long-range phenomenon. So because these chunks of chromosome are so big in meiosis that get inherited together, you can detect linkage over very long genetic distances. And what that meant was that we could do a linkage scan, a genome scan across the whole of the genome with a very small number of markers. So you might have 400 markers, spread them across 23 chromosomes, and you would have a reasonable chance that one of them would be close enough to your disease locus that it would pick up linkage, even though you had no idea where that disease locus was. By contrast, association is a very short-range phenomenon. You will only see association if the marker variant is absolutely right on top of the gene of the variant that's actually giving you disease. So what that means is two things really. One is that association is giving you a closer localization, so it may be more useful from that point of view. But the other is that potentially you either need to know where to look in order to try to detect association, or if you want to do association across the whole genome, you've got to use a very large number of markers. So people typically use 1 million markers to try to capture the amount of variation that there might be in the genome. And what we could discuss, but we won't here, is that that kind of association only picks up very common variation. It doesn't pick up rare variants which might be causing disease. But if you want to pick up all the common variation in the genome, you need around, say, you've got to have about 500,000 or 1 million or so markers. So how things were done, so was that people, the original concept of mapping disease genes was that you would carry out linkage studies, you get an approximate localization, and then you have some very densely spaced markers in that region implicated by linkage, and you'd hope to detect association, and you get stronger and a stronger association, and that would give you that would eventually take you to the disease gene, and that was successful for say a large number of diseases. But then, and nowadays, if people do a genome-wide association study, then you'll need 1 million markers, and this is the typical. This is typically how the results of genome-wide association study are summarized. This is called a Manhattan plot, and I'll just take you through it so give you an idea of how to interpret these. So along the x-axis, we put out all the chromosomes, one to the X chromosome, and so each dot is a different SMP marker. So notionally, there's about 1 million dots in this graph. The x-axis gives you the position of that marker on along the chromosomes, and the y-axis is the statistical significance of the association that's observed between the marker and the disease. So typically what you do is you take a few thousand cases, a few thousand controls, you'd see whether the marker is, that one of the allele of the marker is commoner in the cases or commoner in the controls. You'd do a statistical test, a chi-square test or a logistic regression test, and you get a p-value out which says how statistically significant is the association between that marker and the disease phenotype. Then what you do is you take the logarithm of that p-value and you take the negative of the logarithm. So if I have a p-value of 0.01, p is 0.01, then that is a p of 10 to the minus 2, 1 over 100. 10 squared is 100, 10 to the minus 2 is 0.01. So a p-value of 10 to the minus 2 is p is 0.01. The logarithm of 10 to the minus 2 is minus 2. So the logarithm of p is 0.01 is minus 2. And the minus log p is then 2. So if I have a p-value that's 0.001, that's 10 to the minus 3, then log is minus 3, MLP is 3, 10 to the minus 6 is the MLP is 6, 10 to the minus 9, the MLP is 9, and so on. So what we're plotting here is the minus log p-value, and because this line here is the line for overall statistical significance. So what we're doing here is we're doing 1 million or so different statistical tests, and that means that if we had an ordinary p-value of p is 0.05, then that's 1/20th, probability 1/20th that we'd observe this data by chance. Then if I do 1 million tests, then the number of positive statistically significant tests I'd see would be 1 million over 20, which is a very, very large number indeed. We'd have loads and loads of statistically significant tests. So what we do is we adjust the significance level so that we to 10 to the minus 8, 7 or 8ish or whatever, somewhere in that region, which depending on exactly what our assumptions are about how independent all the SNPs are. But the meaning of that statistical significance is we expect that by chance, even if we do 1 million tests, there will only be a probability of p is 0.05 that one of them would reach that level by chance. So then everything above this line is called genome-wide significant, genome-wide statistically significant. And what you can see is there's a whole bunch of SNPs, 100 or more, where the association with the phenotype of schizophrenia is stronger than you would expect by chance, even though you've done 1 million different tests. Okay, so that's a Manhattan plot. There's kind of variation that's just helpful to have heard about because it gets to be important in different diseases, and this is just to explain why it's called a copy number variant. So there's a kind of variant called a copy number variant, where if you imagine you've got 2 chromosomes, and I've just color coded them here, the regions, And so because you've got 2 chromosomes, then for each bit of the chromosome, you have two copies. If you delete a bit of one chromosome, then you end up with only 1 copy here of the red and the blue bit. And if you duplicate a bit of chromosome, you have 3 copies here, the orange and red. So copy number variants are just places in the chromosome where you've either had a duplication or a deletion. So what we're kind of getting more towards nowadays is, following on for those mapping studies and genome-wide association studies, is sequencing. And sequencing is a technology which used to be incredibly expensive and difficult. There was a technique called, which is still used, called Sanger sequencing, which is very reliable, very, very accurate, very expensive and difficult. So if you want to do sequencing, what you would do was you'd have to know very precisely which bit of the genome you were sequencing, which bit of the gene, and then you look very intensively at that, and you might detect some variation. But sequencing means you're actually reading off each base one at a time. You actually read the whole genetic code. Then what's happened over the last few years is it's become possible to do whole exome and whole genome sequencing. So whole genome sequencing means that what you essentially do is you get, as you're well aware, you get the entire DNA sequence, 1 base at a time, of all 3 billion bases. Exome sequence is the same, except you only look at the exomes, the coding region, so that's 1% that's 1% of the sequencing that you need to do. And when this was done originally, it was again extremely difficult. It's a massive undertaking to carry out the to sequence the whole human genome. And nowadays it's a kind of absolute routine exercise that basically costs $1,000 or so to get the whole genome, or it might be less nowadays, and is done high throughput and is kind of pretty routine and very, very it's very, very accurate. It's not quite as accurate as Sanger sequencing, but it's very, very accurate. So we can read the entire sequence of every gene we want to, and or if we want to, the whole human genome. So just to sort of fill in a little bit about where genetic studies get you to. So if you get to the stage where you've got a variant, you found the gene, you found a variant, you think that variant is producing a particular phenotype, then generally you want to take that forward to kind of prove it and understand it better. And then there are various ways that you might want to investigate it further. So one thing is you might want to see whether it affects the amount of RNA that the gene is producing, which is called expression studies, and that's kind of done very routinely now with RNA-seq, just measuring how much RNA, messenger RNA, there is corresponding to different genes. You might want to look at the protein which gets coded for, how much of it there is, where it is, what it does it carry its enzymatic properties properly, is it immune to particular control mechanisms, is it switched on when it shouldn't be, is it impossible to switch off, impossible to switch on, stuff like that. You might look in cell cultures, and you might look in a whole animal. You can have animal models. You might look in a fly, a zebrafish, a mouse, a rat, what have you, and so you can, to be clear, you can, once you have a DNA variation which you think might be associated with the disease in humans, you can take that variation, you can put that variation, or you can put that whole gene, or you can put just the variant that you're interested in into the animal gene, and you can see what effects that variation has in the animal. You can switch it on in particular tissues, you can switch it off in particular tissues, and you can do all sorts of astonishing stuff in terms of trying to understand, when you have a DNA variation, what the effects of it appear to be. So to come back to genome sequencing, just to give you a rough idea about it, and I think I want to show you something which is that I think when sequencing was introduced, I think we had the idea, oh, this is going to be great. We'll just read the DNA sequence and we'll be able to see what's wrong with it, and we'll take people with the disease, see what's wrong with their genetic code, and then we'll understand the disease. So what you're doing, you've got 3 billion bases. I say it costs maybe $1,000, and there's in humans around 20 something thousand genes. So this is the amount of DNA variation that you get when you sequence somebody. So you've got the genome first, you've got 3 billion bases. There's, say, 3 million very common variations where it's very common to have variation between one person and another, one chromosome and another. There's 3 million places in the genome where there's kind of common variation, and there's 500,000 of that is common little indels, insertion solutions, where a little bit's put in, a little bit's taken out, so sort of minor variations in the genetic code. And when you look to see whether you think a disease is causing, sorry, you can't see the bottom line of that, I guess, but when you want to see whether you think something's causing disease in somebody, what you have to bear in mind is that each individual person will have 100,000 or more different places on the genome where they've got a variant that you've never seen before in anybody else. And of those, a number of them will affect function, which we can't see at the moment, but we'll come to the exomes for that. So when you're looking at the exomes, you've got 30 million exomes, so you're going to say, right, well, there's a whole lot of stuff going outside the exomes that I don't really care about, but at least if I look at the exomes, I should be able to understand what's going on. And again, what you see is you have thousands and thousands of places in the exome where you have common variation, which is so one amino acid get exchanged for another. You've got insertions and deletions with amino acids coming and going. You've got thousands of non-synonymous variants where the amino acid sequence is different between people, of which maybe 1,000 or 2,000 will be very rare and unique. So you will look at it and you will not have seen that before in anybody else, and you will not know whether that variant that you're looking at in that person, say with that disease, has anything to do with that disease or not, because you've never seen that variant before in anybody else. And so you've got many hundreds of variants which are likely to affect function, and when you get the results of DNA sequencing and you try to match that up with somebody's phenotype, the disease they have, or in association studies with disease, it's a very challenging task. So just to again emphasize that, so this is me trying to do association of in a few hundred controls and 1,000 or more cases, and this is just the variation in one gene. So each line here is a variant. It's in different places, intronic, synonymous, there's a non-synonymous variant there. And basically you have all this variation, and you're wanting to say, how does this variation in sequence relate either in a large sample of thousands of case and controls with risk of disease, or in one individual patient who's got a new rare disease? Is it causing the disease I'm interested in or not? So it's very challenging, and people are struggling to deal with this sequence data. Okay, so that was all that I wanted to say, really. I'll just a kind of taster of the fundamental concepts of genetics. I go into more detail in all this in the accompanying handout, the notes. Genetics is very complicated. There's an awful lot of it. There's a lot of bases and a lot of variation, and there's just a lot. Hopefully I've got across the key concept about inheritance and meiosis, and the way that big chunks of chromosome tend to get inherited together, and that is important in the way that it underlies linkage. And I realized I didn't make it explicit, but the idea about association and linkage disequilibrium is that if we go back many generations, and we have these big chunks of DNA that have been inherited together, they will get broken up by crossovers over time down the generations, and you end up with smaller and smaller fractions of fragments of DNA where a disease mutation and the nearby variants are still together, and that process is called linkage disequilibrium. So that the phenomenon that underlies association is that you can have variants which are close to each other, which haven't been separated by crossovers if they're very close together in very tiny fragments of DNA. So that meiosis is central to that process of accounting for association, which is preserved over generations. We can think about genetics in different ways, and it's commonly spoken of loosely in all sorts of different ways. So hopefully to make that explicit, and you'll come across that in different places. Here I've talked very much about the biology of genetics. And I mean, just to finish, to say, really, I think it's given the huge complexity, it's actually quite amazing that it is possible to identify disease genes, find associations with particular genes, find particular variants in those genes which are associated with disease, and then model those and understand how a DNA variant affects ultimately the functioning of a cell and produces effect on the phenotype. Okay, so with that mildly positive comment, I will finish and wish you luck with the rest of it.